It's well known that plants need nitrogen to grow. Nitrogen is a key component of the backbones of DNA and amino acids, which make up the proteins that give plants their bulk. But plants can't just take nitrogen straight from the air. That's because atmospheric nitrogen is in the form of N2. This form of nitrogen is locked together in a triple bond that takes an enormous amount of energy to break apart. Some soil bacteria can convert atmospheric nitrogen into reduced forms like nitrate and ammonia, which plants can absorb through the soil in a process called nitrogen fixation. Leguminous plants like peas and beans house these nitrogen-fixing bacteria in nodules on their roots, giving them a consistent supply of reduced nitrogen. But this isn't the case for cereal crops like wheat and corn, which make up the bulk of the world's caloric intake. So crop growers often add synthetic fertilizers to give cereal crops the nitrogen they need. While this does help plants grow, chemical fertilizers can leach from the soil and end up in aquatic ecosystems through nitrate poisoning and algal blooms, which deplete oxygen concentrations in water. One proposed solution to reducing the need for chemical fertilizers is to take key genes from nitrogen-fixing bacteria and transferring them into plants so they can fix nitrogen themselves. However, nitrogen fixation in bacteria requires a large gene cluster called the NIF operon, which is strictly regulated by embedded layers of activators and inhibitors. Plants, on the other hand, do not organize their genes into operons, so simply transferring the NIF operon into a plant genome wouldn't work. To complicate things even more, the metal cofactors of the nitrogenase enzyme is sensitive to oxygen and requires a lot of energy. The plant's chloroplasts have been identified as a promising target to transfer these genes into. Chloroplasts and mitochondria are the remnants of ancient bacterial cells, so they have similar gene regulation mechanisms, and their genes are held in circular DNA similar to bacterial plasmids. And they consume large amounts of oxygen and contain lots of ATP, satisfying the redox requirement of nitrogenase. To make the NIF operon compatible with plasmid DNA, a team led by Chris Voigt refactored the NIF cluster to remove its native regulation and replace it with synthetic genetic parts to simplify regulation in a plant host. The first step was to remove all non-coding DNA and native regulatory genes. Then the NIF operon was codon optimized to resemble the codon usage of a eukaryote host. This changes the nucleotide sequence of each codon without changing the amino acid. So to gain some control of the system's expression, different NIF genes were encoded into synthetic operons, each with their own promoters, so that the genes are expressed at appropriate levels. Different sections of these synthetic operons needed to be expressed at different levels. So there were combinations of weak promoters, medium strength promoters, and strong promoters. Synthetic ribosome binding sites and spacers, as well as terminators, were also incorporated into this synthetic operon cluster. A controller module consisting of genetic circuits and sensors feeds into the synthetic operon and regulates the conditions and dynamics of gene expression upon the introduction of chemical inducers. The controller is activated in the presence of the inducer IPTG and in the absence of N-hydrotetracycline, enabling precise control over the expression of the synthetic operons. The controller module replaces the native regulation by NIF-L and NIF-A, which were taken out of the refactored cluster. The final product is the recoding of a large gene cluster with complicated regulatory feedback loops into a modularized set of synthetic operons consisting of standardized biological parts, enabling further debugging and improvement. In following years, the refactored NIF operon was further modified and successfully expressed in a eukaryotic host, Saccharomyces cerevisiae, for the first time. This marks a key milestone towards the expression of nitrogenase in plant hosts. While this is a huge breakthrough, further work is currently underway to finally achieve expression of the bacterially derived NIF operon in the roots of cereal crops for the first time.